Alright, in this video, we'll be looking closely into the process of translation. Well, translation is where the mRNA gets translated into polypeptide. Okay, in the previous video, we have discussed how the transcription process takes place. And now once the mRNA is completed inside the nucleus, okay, the mRNA is formed inside the nucleus. Okay, it is to leave the nucleus through the nuclear pore, making its way into the cytoplasm, and off they go into the ribosome. Okay, so it is in the ribosomes where the translation process will take place. Alright, before we discuss further the process of translation, okay, we need to discuss some necessary information that the translation involves. Okay, so first, the translation involves the ribosomes. Okay, now our ribosomes are made of two subunits. Okay, we know large and small subunits. Alright, and secondly, ribosomes have these three domain sites. Okay, they have these three domain sites known as the E, P, and A site. Okay, the E, P, and A sites. Okay, these are the three domain sites in the ribosomes. Okay, and ribosomes are 70S in prokaryotes, 80S in eukaryotes, and some of these ribosomes are bound on the endoplasmic reticulum. Alright, so this is the ribosomes. Secondly, okay, the tRNA molecules, okay, they are single-stranded, okay, and tRNA molecules can range from 75 to 95 nucleotides in length. Alright, and one thing unique about tRNA molecule compared to the other RNA molecules is that they fold into a cloverleaf configuration, which gives it a 3D structure. Alright, so this is the cloverleaf shape of the tRNA molecule. Alright, which gives it a 3D structure. So it folds. Okay, so this single-stranded tRNA molecule folds, okay, into this cloverleaf configuration, which gives it a 3D structure. Okay, and the tRNA molecule has a specific amino acid attachment arm and the anticodon arm. Alright, so this is the amino acid attachment arm and the anticodon arm. Alright, okay, so let me explain that this is where specific amino acids are attached. Alright, and the anticodons, the anticodon arm, is where there are specific triplet bases found here. Okay, so the anticodon arm is where there are specific triplet bases are found. Okay, now let me emphasize this very strongly. Each tRNA molecule has a specific set of anticodons. Alright, every tRNA molecules have a specific set of triplet bases here. Alright, and this correspond to the type of amino acids found here. Okay, so we will discuss about this formation of activated tRNA molecule, or this formation of amino acyl tRNA molecule. So activated tRNA molecule, amino acyl tRNA molecule, they simply mean that this is tRNA molecule attached to a specific amino acid. And you must know that this is an enzyme specific reaction, okay, where one amino acid, each amino acid is correspond to one specific anticodon. Alright, so this enzyme amino acyl tRNA synthetase facilitates the joining of specific amino acid to a specific tRNA molecule. Alright, so you must know this very well, and we be very clear about this, that each tRNA molecule must carry a specific amino acid, and this must correspond accurately to the anticodon that they carry. Alright, so this is how the amino acyl tRNA is formed. Now let's look into the translation process. Okay, first, this is the ribosome. Alright, and when the mRNA leaves the nucleus, alright, into the cytoplasm, alright, it enters the ribosome. Okay, the mRNA enters the ribosome, and what the mRNA carries are a set of codons. Alright, this is what we call as codons. Okay, so what happens first? Alright, so what happens first is that there are specific tRNA molecules that must correspond to the codons here on the mRNA. Okay, so let's work with one example. Alright, so the first example is the tRNA molecule that carries the anticodon UAC. Okay, now you see these anticodons UAC, okay, correspond to this amino acid called methionine. Alright, so as I mentioned just now, in the formation of amino acyl tRNA molecule, each amino acid must correspond to a specific anticodon. This is how the amino acid is attached to the tRNA molecule. So you see, in this case, methionine correspond to UAC, alright, and therefore methionine gets attached to this specific tRNA molecule. So what happens here is that this tRNA molecule gets into the P site, alright, and the tRNA is bound, okay, into the P site because the anticodons complements the codon sequence in the mRNA. Alright, so this calls for this specific tRNA molecule to occupy the P site of the ribosome. Alright, as you can see here. Alright, and then what happens next is another specific tRNA molecule with the anticodon CCG, alright, that correspond to this amino acid called proline. Alright, so what happens here is that these tRNA molecules get to enter the A site of the ribosome and gets attached there because again the anticodons complements the codon in the mRNA molecule. Alright, so well, in the first step, this is what happens. Specific tRNA molecules enters the ribosome because the anticodons complements the codons in the mRNA. Okay, so therefore these two tRNA molecules get to occupy the P and the A site of the ribosome. Now, remember this very well. Whenever the P and the A site of the ribosome is occupied, alright, we have this enzyme called peptidyl transferase. Now what it does, peptidyl transferase, is to catalyze or to form peptide bonds between these two adjacent amino acids. Can you see this? Alright. So in this case, methionine gets detached from its tRNA molecule, okay, and it's bound to proline instead. Okay, so this is catalyzed. This reaction is catalyzed by peptidyl transferase. Okay, so again, alright, whenever the P and the A site of the ribosome is occupied, right, okay, peptidyl transferase will catalyze the formation of peptide bond between the adjacent amino acid. Okay, then what happens next? Okay, what happens next is the ribosome that will slide. Okay, you see the ribosome will slide along the mRNA molecule, and once it slides, can you see that now the A site is unoccupied? Alright, the A site is unoccupied, and while the E site is now occupied by the first tRNA molecule. Now what happens here is that this tRNA molecule will now leave the E site to be recycled. Alright, this is where they get to be attached again to their specific amino acid. Okay, so here the tRNA molecule leaves, and what happens to the A site is where they are ready to accept new tRNA molecules. Alright, so the corresponding tRNA molecules is the one with the anticodon GCU, and anticodons GCU correspond to this amino acid called alanine. Okay, and this alanine, okay, these tRNA molecules will move into the ribosome and gets attached there because why? Because the anticodons complements the codons in the tRNA molecule. Okay, so what happens again, okay, as what I've described just now, whenever the P and the A site of the ribosome gets occupied, alright, okay, whenever the P and the A site of the ribosome gets occupied, peptidyl transferase will stimulate the formation of peptide bonds between the adjacent amino acid. Alright, so in this case, proline now will lose the attachment to the tRNA molecule, and instead it gets bound to alanine. Alright, and from here on, the ribosome shall slide further and move on to repeat the process until the amino acid sequence or the amino acid chain is complete.